In biopharma, waiting weeks for microbial test results can cost millions. Our rapid automated testing is 50 times faster. By accelerating contamination detection, we're not just saving time and money, we're helping the industry bring safer products to patients faster. That's not just a bold statement, it's a direct challenge to decades of microbial testing. And today on Life Sciences Decoded, we're unpacking that quote with the man behind it, Dr. Jack Regan, CEO, CTO, and co-founder of MantaBio. Welcome to Life Sciences Decoded. I'm Megan O'Sullivan, your host, and today my guest is Dr. Jack Regan, CEO, CTO, and co-founder of MantaBio, a company tackling one of the biggest bottlenecks in biopharma, slow microbial detection. His work bridges cutting-edge diagnostics, automation, and real-world industry needs. Jack, welcome to Life Sciences Decoded.
Thank you very much, Megan. Appreciate it.
Great. Before we dive in, can you tell me a little bit more about the industry so our viewers have some perspective as to the problem you're solving?
Yeah, absolutely. So we're focused on the biopharma industry. When people think about pharma, there's really actually two aspects to pharma. There's the, if you will, pressing of pills aspect of pharma, as well as biopharma. So what is biopharma? Biopharma is the process of manufacturing a biologic. Examples of biologics are things like therapeutic antibodies. You've probably heard of Keytruda. Keytruda is a blockbuster drug manufactured by Merck. It targets numerous different cancers. We also have Humira, another monoclonal antibody manufactured by AbbVie that targets autoimmune disorders. So these therapeutics are actually manufactured in very nutrient-rich bioreactors. And these nutrient-rich bioreactors are also warm. They can become contaminated with microbes. And this is the exact industry that MantaBio is targeted, trying to improve the testing capabilities of biomanufacturers.
It's a problem that I would imagine is very costly. And according to your quote, if I were to decode it, essentially you're saying waiting days for kind of the microbial results during this upstream or downstream process has a huge impact on cost and the time to market for biopharma.
It absolutely does. I mean, these blockbuster drugs, when they're manufactured, the value of those drugs is really, really high. The largest bioreactors out there are 20,000 liters in size. If you equate that to dollars, street value dollars, it's roughly, $20 million of product that is at risk when they're manufacturing that drug. Now, in manufacturing the drug, if it's taking a minimum of 5 days, up to 28 days to get results back, you're operating in the dark. You completely are blind to the fact that there could be a microbial contamination present that you're not aware of. You're detecting it 28 days later. During those 28 days of time, potentially, that problem is ballooned into an even bigger problem that's even harder to solve. Not only are you going to be risking the $20 million of product in that particular line, but if the root cause is due to a contamination in a raw ingredient, multiple lines may be affected. Easily, these contamination instances can result in over $100 million in losses for a company. So it's a very, very big problem that they're looking to solve.
Wow. So essentially, without the real-time data that MantaBio provides, biopharma is flying blind and potentially losing millions.
Absolutely. I mean, they're looking to help mitigate their risks, allow for more reproducible, if you will, manufacturing. If they're going along and all of a sudden they have a contamination event, the line has to be shut down, potentially even the entire plant for cleaning up, right? During this entire time, lost opportunity for selling drugs. And God forbid the product gets out onto the market and there's a recall, you have potential FDA fines. There's a lot of downside to not catching a contamination early. And so the only way to improve the ability of a manufacturer to produce the drug and have it be safe is to increase the frequency of testing and the speed at which the tests are actually executed. And that's exactly where MantaBio comes in. We're developing a technology that can screen these samples coming off of the line and provide results in just 2 hours rather than waiting a week to 4 weeks for results to come back.
So tell us about the Manta Vision system. I'm sure that's what you were just describing, but is it revolutionizing microbial detection compared to the conventional methods that are out there? What's the game changer here?
Yeah, so let me sort of, at least describe the conventional method first, and then we'll go into Manta Vision. The conventional method is cell culture. Cell culture has been arguably known about and used for over 100 years. It's a process of taking a sample out of a bioreactor or any sample, plating it onto a petri dish, putting that petri dish into the incubator, and waiting for growth. You're waiting for colonies to emerge. Colonies are just collections of bacteria that have multiplied over the course of time such that they can become visible to the naked eye. Because different bacteria grow at different rates on different types of media, you have to wait an awful long time in order to confirm the sample is actually free and clear of contamination. So this is considered the compendial or the conventional method. It's very, very slow. And because of this slow speed, the industry is looking for faster tools to enable them to do better risk mitigation as they're manufacturing a drug. Certainly with the invention of PCR has been widely viewed as a gold standard chemistry that allows for a very, very sensitive detection of genetic material. And what MantaBio and the Manta Vision system is doing is we are fully automating sample prep followed by PCR. But specifically what we're doing is we are tailoring and building this technology specifically for the industry. This is not a technology that is being adapted from a clinical purpose into biopharma. It's being specifically built with biopharma in mind. So how is it different? With biopharma, you're looking to detect microbes at the earliest case possible so that you can nip the problem in the bud. You don't want to be finding out a week later or 4 weeks later. You want to find out hours later. And the only way to do this is to try to get yourself around the problem of what we call subsampling error. Subsampling error just means that if you have very, very few microbes, if you go grab a sample out of the bioreactor, possibly you're going to miss it just because there's so few around. How do you prevent missing it? You actually process a larger volume of sample to statistically improve your chances of detecting it. So our system is designed to process a larger volume of sample, which is very, very unique. No other company does that. Fully automated to do high quality gold standard sample prep. Sample prep is just a process of extracting and purifying genetic material from a sample because the quality of the genetic material going into PCR really does matter. And we follow up the sample prep with a larger than typical PCR reaction because, again, you are driving towards sensitivity. The end goal here is not to compromise and take any shortcuts, but to get the highest quality, most accurate data possible for the manufacturer to have confidence that any signal they see is real and needs to be taken seriously.
So essentially, it's not just the cost efficiency, and the time to market, but it's what you need for interventions to stop things before you've spent all of this time in a process that is now the data's gone and the data's not accurate.
What the industry is looking for is they're looking to effectively stop operating in the dark. Right now, when they manufacture a drug, they are effectively blind to what's happening in the bioreactors. As a consequence, if there is a contamination, it's a very, very big problem. It's costly to recover from. So how do you avoid that scenario? You go to faster tests, so you're not waiting weeks. 2 hours later, you're getting sort of effectively near real-time data on the bioreactor. And importantly, if you detect something, you've detected it very, very early on. So there's very, very little contamination present. Potentially, you can, mitigate that and sort of preserve the batch, if you will, and continue manufacturing. Or you just choose to scrap. But importantly, you've caught it so early, you haven't lost weeks of manufacturing time. You can sort of restart. You haven't lost a lot of the opportunity in actually manufacturing that drug and getting it to market.
I've seen on the website that the footprint of this Manta Vision system is pretty small. It's not huge. It looks like it could fit anywhere. Is there an advantage to that, or is it just more cost efficient? What's the idea behind that?
Size certainly does matter, but in this particular case, it certainly isn't the leading factor that determines whether or not a company adopts the technology. Far and away, data quality is going to rule the decision. And so MantaBio with Manta Vision, our primary concern right now is just highly, highly accurate data, reproducible data such that the end user, the biomanufacturer, has confidence in the data. That far and away is more important than size. That said, size is important. They're looking for systems that are small, that are affordable. Why they want the small and the affordable is because they like redundancy, they like having the capability to scale testing if needed. For any particular manufacturing line, All the research that we've done indicates that on average, manufacturers want to place 4 Manta Vision systems on a line, and importantly, have one in reserve should one all of a sudden have a mechanical issue, which we certainly hope will not be common, but nonetheless, they want one in reserve. Having these sort of effectively 5, Manta Visions servicing a line gives them the screening capability to have a constant, if you will, heart rate monitor on the bioreactor at every stage of manufacturing. Now, when you manufacture a drug, it's not like you go from point A to point Z and it's done. It has to be scaled up in size. Then you actually have to harvest. Then you have to go through and purify and effectively polish the drug before it's put in a vial and sent off to the patient. During every phase, as I said, the 4 discrete points at which these manufacturers want to test, they want to have the peace of mind that it looks good at stage 1. They can monitor it again at stage 2 to make sure it still looks good. If at any point they see a red flag pop up, they can sort of pause and say, okay, is this real or not? Allows them to, again, identify problems early, lessen the financial risk of having this 20 to $100+ million loss, and instead have something which is much more manageable where, yes, they may be losing a couple of days, but importantly, they're not losing weeks. And I should say that the number we have heard from the industry is every day the manufacturing line is down, it's at least $1 million of lost opportunity. And when the line is down, that's just the fact that they're not manufacturing anymore. Never mind the fact that they had a drug that they've had to actually scrap. But the fact that they're out of production, it's a big deal for them. They're very eager to get back up and running. They want a small modular system to be able to be all hands on deck. Let's do all this testing because they need to get to root cause analysis. Understanding where the contamination came from, tracking that contamination is extremely important. And this gets into a second value add for our technology. So our technology, not only does it do very broad spectrum testing, it can be equipped with reagents to do what we call speciation. That is the ability to dive into a sample that you've determined to be positive for, say, bacteria, and provide the system a test which is specific for the species detected so that you can confirm that that particular risk is now fully taken care of, because you wouldn't want a scenario where all of a sudden you have 2 contamination events happening, but you can't actually distinguish the 2 from one another.
That's a lot to unpack.
Sorry.
But what I love the analogy of it being the heart monitor of your experimentation. I mean, that is just in your whole manufacturing. And that kind of ties it into the nice size footprint, because when you think of a heart monitor, it's sort of a small, device that tells you a lot. And that's critical. I do want to ask a little bit about, the real world deployment of the system. You're currently in some biopharma manufacturing facilities, you're testing it. Who are you working with? Who do you want to work with right now? I mean, you have this whole new opportunity. Who do you want to be speaking to?
Yeah, so I want to be clear that as of today, we do not yet have products in the field. We are still developing our technology. So this is going to be some time before actually into customers' hands. That said, we've done an awful lot of voice of customer. We have some very close ties with some very big biomanufacturers. They are very interested in working with us. They are very dissatisfied with the current testing solutions they have available to them commercially today, and they're looking for newer technologies. Through this voice of customer, they have effectively defined exactly what they're looking for, and our technology is being built specifically for them to meet their needs. So it's important for us to continue these relationships, because really what's going to end up happening is once the system's ready to go, we need to go through testing and validation before the system is implemented within manufacturing. And so right now, these relationships are being fostered such that we are clear on their needs and we're meeting their needs, and we're going to get to market as quickly as possible.
So I want to do a myth buster. What is a myth about PCR detection technologies that you'd like to debunk?
Yeah, so PCR has this, perception, and it's true, mind you, that it's very technical. And because it's so technical, it has to be done in a reference lab. That by default means that it becomes a slower test and certainly a highly skilled test. The reality is that there's been a lot of advancements over the past 20 years in regards to fluidics and automation, such that importantly, you can fully automate all this chemistry in a system that is extremely easy to use. We often like to refer to our system as like sort of like the Keurig of molecular biology, rather than sort of putting your pod in to get a cup of coffee. You put your sample in, you get a genetic test result out to tell you whether or not you have a microbial contamination problem or not. But nonetheless, these advancements in fluidics and molecular biology, robotics allow us to take that very, very technical test that typically is only reserved for highly skilled PhD trained people in reference labs, where again, you have to ship the sample to the reference lab. Instead, you have that technology in-house where you don't have to waste 18 plus hours shipping, but instead you put the sample on the machine yourself. Effectively, a non-technical person can run it. You don't need a PhD trained individual, but importantly, you get the same high quality data out, again, due to the advancements, in the chemistry and the microfluidics and the automation. And the debunking really is that now PCR is becoming more tangible for effectively lay people to run and get very rapid tests back, such that they can act on that data and effectively have those decisions affect the bottom line of the company.
This seems like such a huge breakthrough. There must be so much excitement around what you're doing. What do you see next for MantaBio?
Well, we're running at neck break speed, so to speak, to get this, product into the hands of our customers. And that is our focus, while not losing touch with these customers, to make sure that as the industry changes, and again, this is an industry that, right now, there's certainly no dominant player providing molecular testing within biopharma. And it is a new, if you will, application for molecular testing in a new industry. And because of that, we have to keep our thumb on, regulations, how they're changing, making sure that we're meeting the needs of the end customer, so that once we deliver them a product, it's going to be free and clear to go, such that they can use it in manufacturing, help them mitigate their risks, become more reproducible or reliable manufacturers of the drugs. So they're not going to be down in manufacturing for potentially a month or more at a time. And certainly, of course, try to improve the safety of the product going to customers, in this case, patients.
That's fantastic. Thank you so much for joining our podcast today. I hope nothing but the best of results for you and success. It's to me, from my background in life sciences over 20 years, I think this is an enormous breakthrough, and I think you're going to be very successful. So congratulations.
Thank you so much, Megan. Really do appreciate it.